So, good morning, everyone. My name is Marina, and today I'm going to present my master's final project that it's called Assessing Syngnathid Status in the Canary Islands through Fissures and Diverse Perceptions. And that study is into Eurosync project, and we focus on the species that we found in Canary Island that are Hippocampus hippocampus, Hippocampus algiricus, Syngnathus typhel, and Syngnathus acus. This species, as you know, has very unique biological characteristics, but they are also considered vulnerable against some threats as climate change or habitat loss. And however, here in Canary Islands, the data that we have available for that species is limited, which makes it difficult to assess properly the conservation status and to develop some effective management strategies. So, because of this situation, in this study, we address some data gaps in changing marine ecosystems, and we also we include knowledge from fishers and divers because they are constantly in marine environment, and because Syngnathid are, in general, they are poor data species, so we need to apply another methods. Because of that, we have three main objectives. The first one is integrating information on Syngnathid status, i.e. species in the Canary Islands from fishers, divers, and scientific literature, evaluating the perceived status and the vulnerability of this species using methods that are suitable for the situation that we have with limited data, and identifying knowledge gaps and conservation priorities for Syngnathid in that region. To achieve these objectives, we used two methods. The first one was a survey-based methodology that has three parts. The first one was the characterization of the diver of the fisher, that we collect general information about everybody that answered the survey. The second part was the characterization of the habitats where these species are sighted or caught, the trends on the sightings, and the causes of the fluctuation of these sightings. And the third part was the knowledge of these species and the interaction that they have with fisheries and diving. Moreover, we designed the surveys in two ways. One for the people that is able to identify each species, and one for the people that is not able to identify these species, and they answer in general for seahorses and for pipe fishes. And for divers, we only have information about seahorses, and for fishers, we have information about seahorses and pipe fishes. The second method that we have is the productivity and susceptibility analysis, because once we collect all the information from the surveys and from the literature, we apply this method that combines two factors. On the one hand, we have the productivity, that are the parameters of the cycle of life of each species, and on the other hand, we have susceptibility, that measures the potential that the species of the study has to interact with some activities such as fisheries and diving. And by combining that two factors, we can assess the risk depending on the species and on the activity. If we move into the results, we collect 49 surveys in total for fishers and 90 surveys in total for divers. And one of the most important things that we noticed was that the vast majority of the people are not able to differentiate the species, most in the divers surveys. And if we move into the fishers, we can see that the species are rarely caught, and some of the fishers told that the frequency is low, and some of them have never caught neither seahorses or pipe fishes. If we move into the changes in the catches, some of the fishers told that, there's a high level of uncertainty, and some of them thought that they are decreasing. If we move into the divers, we have more or less the same patterns as fisheries, And we can see that seahorses are sporadically sighted, well, the sightings are sporadic, and in general, they involve only one or a few individuals. But if we look onto the hippocampus survey, we can see that there's some people that see more than 10 individuals during their divings. If we move to the population changes, if we look to the surveys in which people are able to differentiate the species, we can see that they are not sure about the changes on the populations. But if we move to the graphs of the surveys that people that doesn't differentiate the species, we can see that they think that populations are decreasing. So with all that information, we do the PSA, and for diving, we only have information about seahorses, and we can see that hippocampus is the species that has the low risk, and Hippocampus algiricus has a moderate risk. That is related because of the amount of information that we have, and because some people doesn't know that Hippocampus algiricus it's here in Canary Island, and they don't know how to identify it. And if we go to the part of the fisheries, we can see that more or less follow the same pattern. Here we have the risk graphs for seine fishing, line, and long line fishing. And we can also see that hippocampus is the species that has the low risk. And in that case, Syngnathus tifol is the species that has the higher risk. And here we have the same graphs, but for traps and trammel nets. And we can see that the pattern is the same. And this highlights that we have to take into account not only the species, but also the nature of the activity that we are evaluating the interaction with. So the conclusions of the study are that the 4 species studied remain poorly understood in the Canary Islands, that they are rarely observed and sometimes misidentified, and their level of risk depends on the human activity that we are studying the interaction with. And this study demonstrates that incorporating knowledge from fishers and divers can be a useful tool, especially in situations like that in which scientific data is limited. And finally, that increasing both scientific research and citizen science efforts will be essential to improve our understanding and to develop some effective conservation measures. Thank you for your attention.